My name is Alexis Thomas. I grew up in Rosedale, Indiana, and went to college at Indiana University in Bloomington, where I graduated with a degree in microbiology. Growing up, I was in and out of doctors' offices and hospitals. I became interested in disease and what makes people sick. This led me to become a human biology major and set a track for medical school. Upon arriving at IU, I heard a presentation on microbiology and was so intrigued by all the different types of bacteria and pathogens that I changed my major that day. Until attending IU, I was never really exposed to the possibility of research as a career path. I began working in a lab my sophomore year and discovered that I enjoyed research. I have always enjoyed solving logic problems and puzzles. I view research questions much in the same way and enjoy the process of scientific discovery. Even though I started research in my sophomore year, it was not until my senior year that I felt I had explored other fields and options enough to be confident in my decision to go to graduate school and do biomedical research. The aspects of biomedical research that I am most interested in are host-pathogen interactions and drug and vaccine development. My goal is to obtain my PhD and then work in an industry or government lab pertaining to my interests. As I decided later that I wanted to go to graduate school, I knew I needed time to learn more about the process and the various career paths that are available. When I discovered the PREP program, I knew it was perfect for me. I am able to do full-time research in a field that I am interested in, as well as interact with graduate students, something that I did not do as an undergrad. The program allows for me to get an inside look at graduate school and has affirmed my decision to pursue a career in research. PREP at UGA was the program I decided to join because of the focus on infectious diseases. This closely aligns with my interests and goals. Currently, I am working in the lab of Dr. Stephen Trent. A focus in the Trent lab is lipid A. My research project deals with this molecule. My research mentor for this project is Dr. Carmen Herrera. Lipid A is the endotoxic region of lipopolysaccharide, or LPS. LPS is a large molecule on the outer membrane of gram-negative bacteria. Lipid A is a key factor in inducing an innate immune response. Lipid A is recognized by the immune system via the toll-like receptor for myeloid differentiation factor 2 complex. Activation of this complex by lipid A expressed on wild-type E. coli leads to a strong immune response, which can cause endotoxic shock. Lipid A is a diverse molecule whose structure varies between gram-negative bacteria. These bacteria contain enzymes that alter the structure by changing the number of acyl chains, phosphate groups, or polar functional groups. The structure of the lipid A molecule expressed by a bacteria determines how strong the immune response will be. The goal of my project is to insert lipid A modification genes from other organisms into the E. coli chromosome. Alteration of the lipid A will allow it to be used as a vaccine adjuvant by producing a milder immune response. To alter the structure of lipid A, I will be cloning genes into a plasmid with an antibiotic resistance cassette. I will use the lambda red recombination system to insert genes as well as delete others. Once a modification has been confirmed genetically through PCR and sequencing, we can isolate the lipid A molecule from the bacteria and visualize the changes through radiolabeling and TLC. My name is Alexis Thomas, and I am a PREP at UGA scholar.